Good afternoon, everybody. It's a pleasure to be here and a privilege to be able to talk to you about what University of Mississippi is doing to facilitate or to be part of this initiative of the Genome Phenome study. So I have no financial conflicts of interest. So the Jackson Heart Study is a single-site prospective cohort study of risk factors of cardiovascular diseases in African Americans. It enrolled 5,300 men and women participants, ages 21 to 94 years, from the Jackson, Mississippi metropolitan area. The recruitment and baseline examination started in 2000. This is a study that involves 3 institutions, the Tougaloo College, Jackson State University, and the University of Mississippi Medical Center, and it's sponsored by NHLBI and NIMHD. The 3 institutions house 5 centers. The University of Mississippi's Medical Center is the home of the Field Center and the Coordinating Center, which are responsible for data acquisition, data coordination, and research coordination. Jackson State University is the home of the Community Outreach Center that conducts community activities to continue to retain participants and to conduct community-based participatory research. It also houses the Graduate Training and Education Center that enhances skills in research in cardiovascular disease and health disparities epidemiology in a select group of scholars. And then Tougaloo College is the home of the Undergraduate Training and Education Center that's responsible for increasing the pool of undergraduate students pursuing careers in biomedical sciences. So the Jackson Heart Study has been conducting longitudinal data collection from its inception, including clinical exams, annual follow-up, interviews of the participants, and surveillance of hospitalizations for cardiovascular events. There have been 3 clinical examinations conducted, the first one between 2000 and 2004, the second one between 2005 and 2008, and the third one between 2009 and 2013. The annual follow-up interview is reaching about 85% of the participants on an annual basis. It ascertains their health status and determines whether there have been any hospitalizations in the intervening time period. The surveillance of cardiovascular events started with surveillance of CHD and stroke from the beginning, and of heart failure starting in 2005. The study has been conducting surveillance for mortality all along. The Jackson Heart Study has also characterized its participants extensively in terms of cardiovascular events, as well as a whole range of risk factors for cardiovascular diseases. Some examples of the types of phenotype data that we have include clinical events, measures of clinical disease, measures of cardiac structure and function, a number of measures of adiposity, including visceral and subcutaneous adiposity, pericardial fat, measures of pulmonary disease, traditional risk factors, anthropometry, lifestyle, and medications. In addition, for a number of measures, we have repeated measures across some of the exams, and some of these include measures for circulating and urine biomarkers, measures of psychosocial indicators, occupational histories, neighborhood and environment measures. The study has also collected genetic data. The study has a number of has extensive SNPs, information on SNPs, whole exome sequencing data from a number of projects, and also it's starting to conduct whole genome sequencing initially on a study of asthma in Central America, in South America, and in the Caribbean, but the Jackson Heart Study is also going to be part of the NHLBI whole genome sequencing project that Vasan mentioned a few minutes ago. The Jackson Heart Study also has a nested family study with DNA available for close to 1,500 genetically informative individuals in over 270 pedigrees that will support examination of genome-phenome associations across different kinds of phenotypes, including cardiovascular diseases and other phenotypes. And it will allow also for segregation and other family-based analysis of uncommon and rare variants. The Jackson Heart Study biorepository includes DNA samples from blood cells of more than 4,700 participants, 3,400 whom have consented to allow sharing of genetic data through controlled access repositories such as dbGaP. The biorepository also includes specimens of plasma, serum, and urine, as well as aliquots of mononuclear leukocytes in cryogenic storage for about 1,500 participants. Access to the repository, to the biorepository samples has to be done through a number of approvals, institutional review boards, laboratory or genetics committee reviews, as well as once those approvals are obtained, then the investigators have to sign a data and material distribution agreement and also adhere to informed consent stipulations. The Jackson Heart Study has contributed data to repositories that can be accessed as part of the CVGPS, including dbGaP and BioLink. In addition, the Jackson Heart Study data can also be accessed via the parent study after approval by the recipient's principal investigators, IRB, and relevant Jackson Heart Study committees, such as manuscript proposal committees or ancillary study subcommittees. And also the data has to be consistent with, access to the data has to be consistent with the participant informed consent. All the Jackson Heart Study data stored at a recipient's PI's institution must have appropriate safeguards to protect participant confidentiality. Now, the Jackson Heart Study has the conventional data sharing mechanisms of manuscript proposal process and ancillary study proposal process by which an investigator can submit a proposal to access the data or materials, and once that proposal is reviewed and approved, then the investigator can request data and proceed with the study. And then once the study is completed, the investigator is expected to return the results of that study to the parent study so that data can be shared with other investigators. In addition to the manuscript proposal and ancillary study proposal mechanisms, the Jackson Heart Study has started another mechanism called the Vangar Center mechanism. And this process by this process, an investigator at a biomedical research institution can submit an application to become a Vangar Center and discuss the alignment of the research goals of that institution with those of the Jackson Heart Study, agree to certain criteria of the Vangar Center, some deliverables that are expected of that Vangar Center, such as manuscripts, proposals and manuscripts per year, and then complete data distribution agreement for the PI as well as for other investigators that are going to be part of the Vangar Center. And once the PI completes all that, then he may or she may have access to the data of the Jackson Heart Study, but with extensive documentation in terms of data dictionaries as well as code for working with the data, for analysis of the data. And this, we think, facilitates the discovery process. It allows investigators to conduct exploratory data analysis and to assess whether the data that are available are adequate for evaluating hypotheses of interest. We think that this is a more efficient process for developing proposals, manuscript proposals as well as manuscripts. Currently, we have 21 Vangar centers around the country working with us. These Vangar centers are busy working on manuscript proposals and manuscripts. Some of these Vangar centers have also have investigators who have become co-chairs of working groups, priority working groups for the Jackson Heart Study, including the ones listed in this slide. And at this point, I just want to highlight a couple of publications that have come out recently from this collaboration. One study evaluated the HDL subclasses and risk of coronary heart disease. This study used data from Framingham as well as Jackson, and involved a team of investigators from Hopkins, Framingham, and Jackson Heart Study. The second example is a study of the sickle cell trait and the risk of chronic kidney disease and albuminuria. that involve investigators and studies from 5 cohort studies. So the first example of HDL subclasses and risk of coronary heart disease showed in the 2 Jackson Heart study and Framingham study that the HDL particles that seem to be protective of coronary heart disease were the smaller, denser particles, or HDL3C. They were consistently associated with a lower risk of coronary disease across the 2 studies and across different regression models. The second example of sickle cell trait and chronic kidney disease that examined data from 5 studies showed that there was an association between sickle cell trait and an increased risk of incident CKD, as well as an increased risk of albuminuria across the 5 studies. So in summary, the Jackson Heart study is a large community-based cohort of African American men and women with a broad range of age groups and socioeconomic status strata, with a high retention rate, extensive genotype and phenotype data, with extensive documentation, ongoing surveillance of cardiovascular events and deaths, and established procedures to facilitate team science. So in conclusion, the Jackson Heart study is uniquely suited for the Genome Phenome study in promoting cross-cohort collaborations for precision medicine and for supporting pathway grants as well as grant challenge grants. I want to acknowledge the Jackson Heart study participants, the Jackson Heart study institutions and centers, the Jackson Heart Research Collaborative, and the sponsoring institutions. Thank you.
Thank you, Dr. Correa. Are there questions for Dr. Correa? Yes.
So what I can do is I can show you what kind of data we have, and then I'll show you the website and the information that we have on inflammatory markers, and you can determine whether we have the information that you need for projects on immune response.
Thank you very much.